this episode of Voice of the Sea, we're researching the deep sea food web as part of Exports, the international research project studying carbon flow from the atmosphere to the deep sea with NASA and the National Science Foundation. Scientists studying zooplankton have discovered that these tiny animals are hugely important to the food web and to Earth's carbon flow. Follow along as researchers take zooplankton samples recently collected from the Atlantic through laboratory analyses. We start off talking to University of Hawaii Manoa graduate student Connor Shea. What are some of the questions that you looked at for the Pacific leg of the Exports research?
One of the big ones in the Pacific was what is the primary resource that all of the animals in the deep sea are utilizing, right? So what is their source of energy and carbon? And then beyond that, we wanted to know how is that food web actually structured? How many levels are there? Who eats who? Who are sort of the important players in moving energy from these like small particles that were an important resource to the larger animals that depend on that energy? The deep sea ecosystem is based on sinking particles, things that are generated in the surface ocean, they sink down to the deep sea, and that's like the food for everyone down there. And one of our main findings was that that community in the deep sea is based on a really small size class of those particles, smaller than we thought would be important when we went there. Our other main finding, besides that, we produced a sort of quantitative understanding of how this whole deep sea community is structured. That food web structure is really important, and we were able to put numbers on how many levels are there and all of these important interactions in that community.
Can you describe for me what these different zooplankton are that are living in this deep sea community? Like, what do they look like?
Yeah, so a lot of the community in the North Pacific were copepods. These are crustaceans. These are like the little green guy in SpongeBob. Yeah, they've got these big antennas and a bunch of legs. They swim around and they generate this feeding current with their legs, and they scan for particles or animals, and when they're ready to eat something, they sort of push it into their mouth and then they keep scanning. So copepods were a huge part of the community there. And we did see some movement. So they would move up and down in the water column, moving to the surface at night and then coming back to depth at day. And then we also saw in the deep sea there copepods that had spent the entire summer in the surface ocean. Then sort of in August when we're there, they start this migration down into the deep sea to sort of overwinter and produce offspring. And so they're carrying like a bunch of lipids and just a lot of material down into the deep sea at the end of the summer. There were also a lot of animals called chitignaths. They're pretty cool. They're pretty weird looking. They're these kind of like long pen-shaped animals, almost like worms, and they're really soft, but they're predators. And so what they do is they sit there and they wait. And then when something swims in front of them, they'll shoot forward and grab it. When you have animals that are coming back down from the surface, these chitignaths are waiting at depth, and they'll eat them and then produce fecal pellets or produce poops, and that's one way that they sort of trap that material coming down from the surface at depth. The North Atlantic is a totally different story. We're capturing what's called a spring bloom there, where we have the production of a ton of phytoplankton biomass. So these are the plants of the ocean, and they're producing a lot of material because there's all this nutrients and sunlight that hasn't been available all winter long in the springtime. You have this community basically of vegetarians, right? And they're just gorging on all of these plants that are being produced in the surface of the ocean. They don't need to be efficient, they don't need to recycle material, they're just trying to, eat as much of this algae as they can. There's no possible way that they can keep up with that. So you have a situation where you now all of a sudden have a surplus of material and a whole community of animals trying to utilize that.
If it wasn't labeled, could you tell an Atlantic sample from a Pacific sample?
If you knew your zooplankton really well, you definitely could. But yeah, the samples look fairly different. The communities were composed of entirely different animals. In the Pacific, we had a ton of copepods and a handful of different types. And copepods were definitely present in the North Atlantic, but the biomass was dominated by different taxa. One thing that we observed in really high abundance were these pteropods. They're basically like swimming pelagic snails. They're these snails that live in the open ocean. They have a little shell and these little wings, and it's almost like they flutter around down there, and they'd move up and down through the water column day and night. We also at times collected large amounts of amphipods in the North Atlantic, which is sort of another crustacean. So yeah, the communities are fairly different in the two places. And if you had a trained eye, you could definitely tell which was which. That's one of the coolest parts about this whole project to me, is that it really highlights the role of these teeny little animals that live across the entire planet, and they're so intimately connected to our climate and how our planet regulates itself, and the, you know, the global temperatures and the weather that we see. All these things are connected, and these little animals that are like nowhere close to where you and I live are having a direct impact on our lives, and that's pretty incredible. Let's go look at some zooplankton. So the samples all live in this freezer when we first get them. At this point, right, they've been collected at sea, and then they've been separated into different specific size classes.
Okay.
They're shipped on ice, so they all show up here frozen. Then it's our job basically to process these samples from whole animals into a powder. This is our freeze dryer. It puts all of our samples under vacuum, and when they're at really low pressure, when they're in the vacuum, the water basically starts boiling off without having to heat the sample. And we need airtight seals here and here, because this whole chamber is under vacuum. And we're going to open this valve, and that's going to suck all of the air out of the chamber where our samples are. So let's go ahead and do that. And inside, there's this coil that's kept really cold. Any water that comes out condenses and freezes on that cold coil. It's right inside the top here. You can actually see it if you look down into it. And you can already see some of the moisture is freezing onto that. The samples will degrade pretty readily if we leave them wet. Ideally, they're frozen when you put them on, and they stay frozen throughout. These will stay on here for about 2 days, and after 2 days, they should be totally dry. So now we have a fully dried zooplankton sample. So we make sure everything's really clean before it touches the samples too, because we need to keep them clean. So we take the entire community here, right? We have all these different animals, amphipods and things. We scrape it into our little dish. And once it's in there, we can just grind it right up. So then, what we'll do next is we'll take our powder and we'll transfer it into this vial, and we can weigh it. So this one is 237.1 milligrams. And that's one sample prepped and ready for analysis.
The University of Hawaii's Sea Grant College program focused on Hawaii's coasts and its communities through sustainable development, safe seafood supply, sustainable coastal tourism, hazard resilience, and healthy coastal ecosystems. Hawaii's Sea Grant.
You're watching Voice of the Sea. We're in the Isotope Lab at UH Manoa, talking with lab manager Natalie Walsgrove as we follow zooplankton samples from the Exports Project through analysis to better understand the deep sea food web.
It's kind of amazing to see how many different projects we are able to be involved with. We've done everything from human samples and what they're eating in African tribes, archaeological samples at Rapa Nui, to fish samples, to birds, to ancient sloths, to it's kind of cool. Lots of algae, lots and lots and lots of algae. I am the manager of the Stable Isotope Facility, and it's my job to just sort of keep the place running. So that involves not only running samples, which I do some of, but mostly what I do is I maintain the instrumentation, I repair it when it breaks, which is often. I train the students in what they're doing, because it's really complicated work and they don't arrive knowing how to do any of this. So I take the time to train them and get them to be able to do these analyses on their own and make sure that they're doing it safely and in a way that will result in high quality data.
How did you come to be the lab manager?
For my master's work, I looked at trophic behavior of ciguatera, ciguatoxin and ciguatera fish poisoning, and I did that work here at the University of Hawaii. And when I finished, Brian Pope needed a lab manager, and so he took me on as lab manager about 10 years ago now.
So we've got these samples of zooplankton, and Connor's just been drying them and grinding them, and they're all ready for analysis. Can you take me through the next step of the process?
Yeah. So this instrument we use for bulk isotope analysis. So when we talk about bulk isotopic analyses, we're talking about all of the nitrogen or all of the carbon that's contained in a sample. Sample that Connor ground up, that's that homogeneous powder, so any small subsample you take is representative of the whole. You would take 0.5 milligram and it gets capsuled into a little tiny bit of tin capsule. That whole thing with the tin capsule and the 0.5 milligram of sample is then added to this instrument, combusted, and then turned into both N2 gas and CO2 gas. There's multiple forms of nitrogen and multiple forms of carbon that are what we call stable isotopes, which is what we do in this lab. And so for nitrogen, it's nitrogen 14 and nitrogen 15. Nitrogen 14 is very common and nitrogen 15 is very rare. And so the ratio of nitrogen 14 to nitrogen 15 is what is interesting here. For every metabolic process that a thing undergoes, the nitrogen 15 will stick around longer. And for carbon, it's the same thing. Carbon has two forms of stable isotopes, carbon 12 and carbon 13. And again, the carbon 12 is more easily metabolized, so it tends to not stick around as long as the carbon 13. So you'll get enrichment with each metabolic process that a nitrogen or carbon goes through. The journey of a zooplankton sample starts in the autosampler up here in a tray like this, where this will sit in the autosampler. And you can see there's one little space where this bottom of this tray is open.
So just one sample at a time?
Just one sample at a time. It'll sit in here and it'll just move forward just a slight tiny bit for each sample when the instrument is ready for it, so that one little cube of that zooplankton tissue will drop down. The job of the EA is just to get that solid sample turned into a gas sample, to turn it from everything that is in some zooplankton tissue into just N2 gas and CO2 gas that are going to come off of this instrument at slightly different times so that the mass spectrometer is able to read one and then the other. Once it's gone through that separation column that'll get the N2 and the CO2 to be away from one another, then it gets introduced to the mass spectrometer, which is the box at the bottom, and that's what will actually measure the difference between the different isotopes, so the nitrogen 14 versus nitrogen 15 and the carbon 12 versus carbon 13. This instrument we use for the amino acid analysis. So that's where we, instead of taking the entire sample, we will isolate the amino acids that are in that protein and then purify them and put them in a form that we can then inject into the gas chromatograph. After it goes to the gas chromatograph, where it separates out each amino acid from one another, it gets introduced to the mass spectrometer, where we can then get the isotope ratio of each individual compound of interest. And for this, it's 14 compounds that come from the sample, plus 3 compounds that are standard, or are reference material, that are injected along with each sample injection. This compound moved the fastest through the column. This one will have moved the slowest through the column. You can see there's nice separation between each of these different compounds of interest. So these are the different amino acids that can be analyzed using this technique. The mass spectrometer is actually reading the isotope ratio of those compounds. That's what's being given graphically here, and then also in a table here is the intensity of the signal, but also the ratio of the 13C to the 12C that's in each one of these peaks.
And what is your favorite part about this work?
Honestly, one of my favorite things is doing the troubleshooting and instrument maintenance, and I love to tinker, and so I get a lot of opportunity to do that here. For every peripheral that is being used, by peripheral I mean like every instrument that gets a sample ready for the mass spectrometer to read, those instruments can't tolerate anything more than a gas. So when you're starting with a little cube of zooplankton tissue, you've got to turn that tissue into a gas before it can ever hit the mass spectrometer. It's complicated to turn a piece of zooplankton tissue into nice, clean, usable nitrogen and CO2. A lot of things can go wrong on the front end. So you take the complexity of both sides of this instrumentation, and it's just, it's kind of a nightmare, really. When we first started doing this analysis 12, 14 years ago, very few labs in the world were doing it, and now it's much, much more common. There's like several labs around the country and the world that you can do it for a fee. So if you have samples you want analyzed but don't have the expertise or the time to do it, you can just send them to them. We do samples like that as well. People will send us their samples and we'll just do all of the preparation, all of the analysis, all of the quality control, and send them back data that they can then use. We've also expanded our capabilities significantly in that time. We started with just one GC doing this work, and now we have 3. So even our own capabilities in the last decade have really expanded.
We are looking for a few heroes, mentors, trailblazers, innovators, a passion to change lives, spark curiosity, open hearts, and awaken minds, help students answer the question, who am I? This could be your calling, but this is no job. It's the journey of a lifetime. Be a hero. Be a teacher.
You're watching Voice of the Sea. We're talking with principal investigators Brian Pope from UH Manoa and Amy Maas from the Bermuda Institute of Ocean Sciences, both lead researchers on the Exports Project, which just returned from collecting samples during a month-long cruise in the Atlantic.
There were satellite observations that were being made that guided the ship to a certain part of the ocean and into this parcel of water in which they hoped they could follow. And that parcel remained coherent through the entire cruise. Even though there were different mixing events, it still remained intact and they were able to follow this.
How can they tell that that water parcel is remaining intact?
The main way that that was done was through making measurements in different parts of that parcel. They kind of knew where the center was, and they could go back to that and drop instruments in and check that. Think of it as like a wheel with different spokes going out. There were 3 ships out there that could do this. While taking samples, they could follow those spokes out to the edge of this eddy and begin to map where that was. The other thing that they had assets in the water, some autonomous vehicles that were programmed to move through that eddy and sort of swim back and forth and take measurements. And in real time, they could get a picture of where this water mass was moving. And it was from all that data, they knew that it did stay coherent. There are what are called mesoscale eddies, large eddies that form in the eastern tropical Pacific that move through Hawaii at times. And one of the goals of the cruise to the North Atlantic was to find one of those coherent eddies and follow that coherent eddy so that we could follow the evolution of the bloom from the start of that bloom when the sun came out and the water warmed to the end of that bloom and follow how the plants change, the phytoplankton change during that evolution of the bloom.
What is it like working with such a large group of really smart people?
Coming at it from so many different directions, we can make so many different connections. We get on a Zoom call with 100 people from around the world, and we're learning so many new things that we hadn't even thought about. And suddenly somebody who is studying something you thought was very, very obscure, they come up with an idea and you go, oh my God, I can see that in my data too. The Zoom meeting yesterday that I was on, I was just mesmerized by this talk about the eddies and how they were evolving and how we can tell the edge of the eddy from the center of the eddy. Like, I'd never thought about that. And how does that relate to where my samples were taken? And how it will relate to other people's observations. The context that we can put our data in is so much more enriched.
I am an associate scientist at the Bermuda Institute of Ocean Sciences. I am a marine scientist. I study zooplankton, so the little animals in the ocean. Exports is monumental. It's the biggest ocean expedition that's happened in a very long time. If you're eating like something that's been eaten 5 times before, the laws of thermodynamics that say you can never have more order is that things are going to get lost along the way. You're zooplankton. You ate a zooplankton that ate a phytoplankton. That's a really short food web. So not a lot of the carbon was lost. Whereas if you're in a different system and you ate a thing that ate a thing that ate a thing that ate a thing that ate a thing, and all the way down to phytoplankton, a lot of carbon was lost along the way. And we're trying to understand that movement of carbon. It helps a lot to have that sense of what the food web looked like. When we talk about a recycling system versus an export system and how that stuff moves and is packaged. And we look at it both in the surface, which relates to the satellite stuff. But one of the other things that's crazy cool about this is trying to understand that midwater food web, because it's understudied and it's really difficult to study. If you have a lot of zooplankton eating in the middle of the water, you'll have way less that makes it to depth. So understanding that food web is really important for bridging the link between what we can see from satellite, which is in the surface, and what we measure coming out at the bottom, right? So we have these traps that look up and they collect particles, and we're trying to figure out what's happening between here and there. And that's all about food webs. So the particles in part are made up of poop, and they're made up of all these little leftover pieces. And we can see traces of the zooplankton in the particles. We can tell whether a particle might have started as zooplankton produced material. And so it's kind of like a fingerprint on where did this particle, which looks like just mush, right? Like, or it's like squished up on a filter, because that's how we sample particles in the ocean. Where did it come from? How much of it came from a zooplankton? How important are they, right? And I can't do that by just measuring how many zooplankton are in the water, because there's processes that contribute to that piece. And the chemistry helps me get at that in a way that I couldn't do.
So then the reverse of that, how does your work help Brian Pope's lab and Connor know about their isotopes?
Yep. So the isotopes, when you've got this signature in the water, they need to figure out how long that signature relates to. How fast could that number of particles be produced? How many would be required? And I give them that ground truth of like, this is how many bugs there are. This is how fast they produce them. And then we can start getting at that middle step of like, what happened in between? You end up with the particles, you start with this, and we can like work towards the center of like, this is the food web. This is how many times this is repackaged. This is what's going on. And again, like you can't, we're inferring, right? We're kind of blindly moving towards each other, but since we're coming from both ends, you have a space to work to try to understand what that food web looks like.
The exports team will continue analyzing results and publishing data for years to come, generating new understandings of the deep sea food web and the global carbon cycle, findings that are critical to predicting future climate conditions. All exports data is free and publicly available. Learn more at voiceofthesea.org. Follow us on social media at Voice of the Sea TV. Mahalo for watching Voice of the Sea. Exploring Our Fluid Earth is the dynamic curriculum developed by the University of Hawaii's Curriculum Research and Development Group. The award-winning Fluid Earth and Living Ocean textbooks are now interactive and online. New activities, updated content, and a teacher community. Exploring Our Fluid Earth is now freely available. Find out more at exploringourfluidearth.org.